Hi, everyone. Good evening. It is my great honor to be here, and many thanks to the CPM for organizing this event, and Nikki for the introduction. So I'm going to tell you a bit more about how we discovered the RNU4-2 variants and also demonstrated that these variants are the cause of the phenotypes of these participants with RNU4-2 or ReNU syndrome. So I think the font is a bit different, but try to explain things. So here we are trying to identify variants in Genomics England participants. So there's lots of participants within the Genomics England dataset which we use for our discovery, and it's quite complicated in looking into these data. So there are a lot of participants. So here I've shown with an example of 5, and then some of them we find variants in non-coding RNA genes. So these are non-coding RNA genes as shown here. And usually they are just identified by what we've been provided with, which is the genome sequencing data that's provided by Genomics England. So most of these variants are inherited from either parent, but some of them are not found in either parent. So these are called what we call de novo variants. So these de novo variants, as we are seeing here, is not found in the father and the mother. So here we see a variant in the father, and then we see a variant in the mother. But in the child, we see an additional variant, which is not identified in either parent. So this we call de novo variants. So these de novo variants are easier to identify and more likely to cause disease. And usually they just come from chance mutations when DNA is replicated in the process of reproduction. So what we've done is that we looked for de novo variants in RNU4-2 or in entire sets of non-coding RNA genes in 8,841 probands, which is the person who has been initially identified with disease in their family with neurodevelopmental disorders in the Genomics England cohort. So among these genes, we looked at all sorts of non-coding RNA genes, including RNU4-2, including many other siRNA genes and other genes with different functions. Normally, as Nikki mentioned before, there is an extremely low chance for a variant to occur on de novo because we see a large size of the human genome is really large with 3 billion bases, but on average, we only see 70 de novo variants in each individual. So this means that on each location, the probability of finding a de novo variant is extremely low. But what we found is shown here. We see a lot of variants being recurrent, being in the RNU4-2 gene in 46 out of the 8,841 probands. So out of all the de novo variants in non-coding RNAs, we see that on the chromosome 12, we have this very recurrent mutation in NDD probands, but we don't see that same mutation in other participants, which includes other probands with non-NDD phenotypes or probands with diagnosed NDD, so they've received a diagnosis, another genetic diagnosis through Genomics England, or unaffected individuals. This immediately led us to suspect that this might be the cause of the phenotypes of these participants. So a closer look into this specific region in RNU4-2 revealed that there is a region that is depleted of variants in the general population. So this shows the entire RNU4-2 gene. And on the top panel here is the patients with NDD that we're finding in Genomics England, whereas on the bottom, we see the population controls. So in the population controls data, we see a region in the middle of this gene that is significantly depleted from variants in RNU4-2. And this region actually matches with where we find the variants in NDD patients. So we've also identified additional patients with the same variants also having NDD phenotypes through global collaboration, including the Gregory Consortium, Undiagnosed Disease Network, SSC, and so on. So these participants in total, there are around 115 we initially identified, and around 89 of them have the highly recurrent insertion variant on nucleotide position 64. So here, this is how we have identified the variants. Then we went on to characterize what we're now calling ReNU syndrome. So this is already named ReNU syndrome. We know that the condition associated with RNU4-2 is now named. And initially, we collected phenotypes data from 49 individuals, and thanks to our collaborators at the Broad Institute. And we characterize ReNU syndrome by a moderate to severe NDD phenotype. So this is reflected by all individuals affected by global developmental delay and intellectual disability, and the majority of them having hypertonia, dysmorphic face features, and speech abnormality. Then we also looked into how the variant in these individuals actually causes this disease. We know that RNU4-2 is the gene that codes for snRNA, or small nuclear RNA. And snRNA is responsible for a function in humans or in all cellular organisms called splicing. Splicing ensures information from our DNA, which is correctly interpreted. So usually on the DNA, which is transcribed into pre-RNA, we have exons and introns. And then the spliceosome comes in. The spliceosome is the mechanism in humans that is responsible for splicing. And it comes in and then cuts out the introns and then puts back the exons together. And we know that the U4 snRNA, which RNU4-2 codes for, is critical in this process. So our hypothesis is that these variants on RNU4-2 actually affect splicing. So through looking into RNA sequencing data of individuals with RNU4-2 variants, we found that individuals with RNU4-2 variants have significantly more incorrect splicing events, as shown here, as we're comparing the controls and the cases incorrect splicing events. So they do have more incorrect splicing events than compared to healthy controls. And this shows evidence that the function of splicing in these participants is impaired. So we show that RNU4-2 variants indeed cause these participants' phenotypes by affecting the function of splicing. So in summary, I've provided a brief overview on how variants on non-coding RNAs can be found in Genomics England participants by a series of variant filtering and annotation pipelines. And then we identify de novo variants on RNU4-2, which is one of the non-coding RNA genes, which is recurrent in NDD individuals, but almost absent in healthy controls. And to test the hypothesis that this variant causes this disease indeed, we showed that these variants lead to incorrect splicing events and thus NDD phenotypes in these affected individuals with ReNU syndrome. And with that, I would like to conclude my talk, and thank you very much for listening.